Okay, good morning, everybody. Today, I wanted to talk about our new in vivo and in vitro single-cell atlas of developing medium spiny neurons. In particular, I wanted to focus on our recent paper where we were able to study the human striatum at single-cell resolution and see how it diversifies from the surrounding cortex and MG. In particular, we combined bulk RNA-seq with single-cell RNA-seq, and this enabled us to find numerous findings. This include, for example, an atlas of de novo long non-coding RNAs and see how they were specific for each area. Then we were able to discover new cell types of the developing human striatum, how these cell types diversify and how progenitors of the LG give rise to medium spiny neurons. Then define the gene regulatory networks that are cell-specific and that are important to establish MSN identity in vivo. And these gene regulatory networks were also validated in silico by doing gene perturbation, so knocking out and overexpressing key transcription factors to predict their function. And then we also found some human-specific long non-coding RNAs by a conservation analysis to see which long non-codings were unique to the human fetal striatum, which makes, let's say, our brain different from other species. These were also validated by fish and immunohistochemistry. So this was the first atlas, our in vivo atlas. And now Paola Conforti has developed a new protocol that mimics human fetal striatum or MSN development in vitro in 2D. And this protocol goes up to 40 days, but by day 25, we have a good number of medium spiny neurons that were classified by immunohistochemistry by DARP32, GAD, and CITIP2. And you can see that by day 25, we have about 20% of MSNs that reach approximately 30% by day 40. We then wanted to see how well this protocol mimicked in vivo development, since we have this in vivo single cell atlas. So we took our human fetal atlas, our new protocol, and performed single cell on our new protocol, and combined the two data sets to see how well or how different the two differentiations are. So this was our main objective, to try and quantify how similar our cells are. So the first step was to classify the different cell types that we have in vitro. So this is our in vivo data. So we have apicoprogenitors, basoprogenitors, this new cell class pre-MSNs that then give rise to D2 and D1 MSNs. And then we have also different types of interneurons in our in vivo single cell atlas. And as you can see, we pretty much have similar cell types in vitro, and we are able to establish, well, obviously apical basoprogenitors, but then that can give rise both to D2 and D1 MSNs. And they reproduce canonical markers. So you can see, for example, ASHL1 in basal progenitors, MACE2 in MSNs, and ILET1 that is specific for D1 MSNs and is not present in D2 MSNs. So this was positive in the sense that we were able to mimic what we see in vivo. If we quantify this, we have a high number at day 15 of apical progenitors and basoprogenitors, and by day 25, you can see that this shifts to a high number of neurons. So we have both D1 and D2, especially D1, it seems to be slightly biased to D1 MSNs. When we quantify how similar the two cell types are, so in vivo and in vitro data, you can see here what you have to look at is at the diagonal. So the darker the diagonal, the better the data is. So you can see here is on the left the in vitro data, and on the top here is our human fetal brain data. And you can see apical progenitors are similar to apical progenitors and less similar to, for example, to basal progenitors. And as you go along the diagonal, for example, D1 MSNs are highly similar to the D1 MSNs and less similar, for example, to D2. This is there are similarities that increases as you go into the neuronal population, obviously. But what we have here is a good diagonal. So this confirms the cell identity that we have in vitro. We then looked at how reproducible the data was. So we looked at the different batches. We have 5 different batches. One batch 2 didn't perform well in terms of cell extraction. So we concentrated on these 5 batches here. But you can see that the maps are very, very similar, and that there are no big differences in cell types. Maybe only batch 3 gives a higher number of D2 MSNs compared to the rest of the protocols, but all of them give D1 and D2 MSNs in the end, the higher proportion of D1s. So overall, we can say that we have an in vitro single cell atlas that we can quantify how similar it is to our in vitro data. And this was not only done by single cell, but it was done by immunohistochemistry, Western blot, and qPCR analysis. And we can show that we can efficiently generate a medium spiny neurons from day 25. By single cell, we see that by day 25, we have approximately 20% of MSNs, and we can get both D1 and D2 MSNs. We show that in terms of differentially expressed genes between cell types with that Jacquard similarity, we have a good overlap, and that our protocol is highly reproducible. So this is a benchmark for future improvements to the protocol, where we can actually study how similar it is to our in vivo atlas. So here is our first starting point, but we can actually quantify at the single cell level how similar the cell types are. So this is it. Thank you for your attention. And if there are any questions, I'll take them.